everyone. Today, my peers and I, Luke Hamilton, Abby Kroom, and Emma Crawford, will be exploring the topic of fluorescence. We'll start with some general information and then take a closer look at some specific applications of the subject.
Okay, so we're going to start with some definitions. The definition of fluorescence is a phenomenon where a substance absorbs a photon and emits a longer wavelength of visible light. This light that's emitted can be any color depending on the substance. A fluorofluor is a fluorescent chemical compound that can absorb photons in the ground state, leading to less energetic photon emission while leaving its excited state. A non-radiative transition is when electrons lose energy but do not emit light. An example of this is vibrational relaxation. One of the most commonly used fluorophores is fluorescein. Its chemical formula is C20H12O5. It has red-colored crystals that, when they fluoresce, they produce a green-yellow light. It is a compound that has a system of aromatic rings, creating a conjugated system. There are many electron pairs to excite in this compound, and the rigid structure makes it difficult to release energy via vibration. So a real-world application of fluorescence involves green fluorescent protein, or GFP. GFP is a naturally occurring compound in jellyfish. Biochemists use GFP to monitor gene expression in cells, so they can tag different cells with GFP and then do gene silencing experiments to see if they're able to silence that gene. They also use GFP to visualize the location of proteins in the cell. The way that we are able to detect GFP in the cell is using fluorescence microscopy. On the left, we can see a schematic of how fluorescence microscopy works, with the sample at the top, and then a light source is going to be applied to the sample. The most important thing about fluorescence microscopy is being able to separate the excitation light from the emission light. So as we know, fluorescence happens once light is emitted, and the excitation light is actually brighter than the emission light. So in order to be able to measure the fluorescence, we have to isolate the emission light. We can do this by using the filter cube, and then as the light goes through the emission filter, all that we're going to be able to see from the eyepiece is the fluorescence. This is going to look like the image on the right, which is cells that have been tagged with GFP and visualized using fluorescence microscopy.
So now I'm going to be going over the discovery of fluorescence, or specifically the history behind it. So the discovery of fluorescence dates back to 1845, and the scientist Frederick W. Herschel was working with a solution of quinine, which is a compound that's found in water. So when light was shined on the solution, it emitted like a faint glow in a sense, and this was the first time that fluorescence was observed. He didn't necessarily know what it was. So a few years later, Sir George G. Stokes, who was a British physicist, investigated the phenomenon more deeply. And in this, he designed an experiment that would help him to better understand this phenomenon. So he used a prism, right? And he separated the sunlight into its full spectrum, essentially a rainbow of visible light. And he directed this light through the solution. And Stokes observed that the quinine glowed blue in areas where no blue light from the spectrum was present. This suggested that the solution was absorbing one type of light, which was most likely ultraviolet, which is invisible to the human eye, and emitting another type, in this case, visible blue light. And from this, Stokes inferred that the emitted light had a longer wavelength and lower energy than the light that was initially absorbed. And he called this phenomenon the Stokes shift, which refers to the difference in energy or wavelength between the absorbed and emitted light. And we can see this in the diagram that is shown to the right of the words on the screen. He also introduced the term fluorescence, which was inspired by the mineral fluoride, which exhibits this glowing behavior under ultraviolet light. And his work basically laid the foundation for modern understanding of fluorescence, which is a phenomenon that is now widely used in fields like biology, chemistry, and things of that nature. So now that we've kind of introduced fluorescence, we're going to be talking about the key steps of this process in a sense, which will be shown in the diagram, right? So we're going to talk about the steps of fluorescence. So first, an electron in the molecule will absorb energy, right? Usually in the form of ultraviolet or visible light. And this energy excites the electron, pushing it from the ground state labeled S0 to an excited state labeled S1, right? So once in the excited state, the electron might initially occupy a higher vibrational level, but it'll start to drop down, right, in that state. And it undergoes vibrational relaxation, that's the term for it, meaning that it loses energy without necessarily emitting light. And this is shown by the red arrows that are labeled non-radiative transitions. So after the electron settles at the lowest vibrational level of the excited state, it'll basically release energy in the form of light, which is called fluorescence. This is represented by the green arrow. We can see as it drops down from that higher state to that lower ground state, light will be emitted. And as it emits this light, the electron will return back to that ground state, right? Basically completing the cycle. So it's important to understand that the emitted light has less energy and a longer wavelength than the absorbed light. So this difference is known as the Stokes shift, like I mentioned before. On this slide, we're going to be talking about the electronic states. So we're going to be looking at two types of electronic states. We're going to be looking at the singlet and the triplet, right? So starting with the singlet, this is when two electrons in a molecule have opposite spins. You can see that illustrated at the top right of the diagram with one arrow pointing up and the other arrow pointing down. And this is the most stable configuration, and that is what we typically find in molecules at room temperature and in their ground state, right? So now if a molecule absorbs energy and gets excited, it can enter a triplet state. So in this case, both electrons have the same electron or spin direction as this, which is shown by this other figure, two upwards arrows, right? So this configuration is less stable and is much less common at room temperature, but it's very important in processes like fluorescence and photodynamic reactions. This lower diagram shows a more visual orbital representation of these two states, again emphasizing the spin difference. So the key takeaway here is that singlet is stable, which is at the ground state, and they have opposite spins, and triplet is excited, less stable, same spin.
Now moving on to phosphorescence. Phosphorescence is a very similar process to fluorescence. It's a process by which electrons absorb energy and then they transform into a triplet state and slowly lose energy by releasing a photon. This differs from fluorescence because it goes to the triplet excited state instead of just the singlet excited state. With the singlet excited state, the electrons are going in opposite directions, but with the triplet excited state, they're parallel, and this is a spin-forbidden transition. This is more common when vibrational levels of two excited states overlap each other. So the steps of phosphorescence are very similar to fluorescence. So the electrons absorb energy the same way and travel to a higher electronic state like S1 or S2. And then these still go through vibrational relaxation. But then these electrons undergo a process called intersystem crossing, where they go into a triplet state and the electrons flip directions so they're parallel. And then at a slower rate than fluorescence, the electron loses energy by releasing a photon and travels back to the ground state. This is the slowest process in phosphorescence. This can take 10, 20 seconds, even up to a few hours. And this is why glow sticks use phosphorescence, because these glow sticks will last for 12 hours before completely stopping their glowing.
And at this section, we're going to go over the Jablonski diagrams. It's sort of a condensed version of everything that we've talked about earlier. It looks a bit intense, but it can be broken down quite easily. So we start over at the S0 state, which is that ground state. Again, from earlier, imagining we have a pair of electrons that then have energy applied to them, either UV or vis, typically. They get excited by something, and as you can see with that blue arrow, they travel up to either S1, which is the singlet state, first excited singlet state, or S2, which is the second excited singlet state. They both still have that oppositional spin as each other. One's just higher in energy. And then they will undergo that vibrational relaxation, go down to the lowest vibrational level. Each one of these smaller little lines that you see is a different vibrational level, while each one of the thicker black lines is one of the electronic levels. So once it undergoes the vibrational relaxation, it can do a couple different things. They can typically undergo something called internal conversion if they're on that S2 level, and then another level of vibrational relaxation to go down from S2 to S1. At this point, they have another couple of options they could do. They can either travel directly from that S1, that first excited singlet state, down to the ground state through fluorescence, as we mentioned earlier, or, and again, all of these options depend on the identity of the chemical. Different chemicals will tend to do different things in different situations. They will undergo something called intersystem crossing. This is probably one of the slower processes here. In intersystem crossing, they're going to travel from that first excited singlet state to something called the triplet state, which, as we mentioned earlier, has the two electrons pointed in the same spin direction. It's a little less stable. And then they'll undergo vibrational relaxation one more time to get down to that ground vibrational state of the electronic level. And at this point, they can either go from that triplet state all the way back down to ground state through phosphorescence, or they can undergo another thing of intersystem crossing, then vibrational relaxation all the way down to ground state. This particular diagram does also tell you the times for how long each of these processes tends to take. So absorbance is by far the fastest, measured in 10 to the negative 15 seconds, which is fast enough to where there's not very much equipment that could even measure how quickly it happens. Every other process is a little slower than that. Fluorescence, for instance, tends to be 10 to the negative 10 to 10 to the negative 7 seconds. Oftentimes it's measured in nanoseconds, which is 10 to the negative 9. Something like intersystem crossing tends to be pretty relatively slower as well, 10 to the negative 10, 10 to the negative 8 seconds. Phosphorescence is typically probably the slowest that you're going to see on here. It can go anywhere from 10 to the negative 6 seconds to even up to about 10 seconds. As mentioned later, a lot of glow sticks and things like that are made with phosphorescence due to how long it can sometimes take to go back down to the ground state. Much of this is attributed to the fact that it does have that same spin state, so then it has to flip backwards to go back down to the ground state where the electrons are against each other again. One other way to visualize this and to go a little bit more into depth with intersystem crossing is to show why exactly the bonds change. So as the bonds undergo more energy, so as it goes from that ground state or that E1 level and goes up to S1 or S2, you see it changes to E2 or E3, it will get higher, but that RE value, which is the distance between two nuclei, so roughly bond length, it's called the equilibrium bond length, stays the same even if it goes up to that excited state. There's nothing really happening with the bond length. But if it goes into the second electronic state, which is that purple arrow represents the intersystem crossing, you'll see something happen with the bond length. With that RE value, it will change. It'll typically get a little bit longer. It undergoes kind of this stretching phenomenon where there's more distance between the two nuclei. So when this happens, even when it goes back down into that singlet ground state, like it will go back down into the ground state like with phosphorescence, it'll still be at a higher RE value than it was before. A longer bond length can create weaker bonds, and it can change the wavelength of the photons that are emitted back out. And this is thought to be why these molecules will absorb, say, UV light at a relatively high wavelength and then spit back out visible light at a lower wavelength. And finally, we do have some math involved in this. So this is a diagram for decay patterns over time. As you can see, this is an exponential pattern. This is going over the intensity or how bright that light is that's produced, the visible light, over time, measured in nanoseconds. This is an example of one used for fluorescence. As you remember, it's roughly 10 to the negative 10 to 10 to the negative 8 seconds. So that 10 to the negative 9 for nanoseconds fits pretty well here. The rate of decay, as you can see, is independent from the concentration. There's no factor for it. You can have one molecule or a million. It's not going to really make much of a difference as to how the decay happens. This means that it does display first order kinetics as well. So this process is still slower than the absorption, as I said, but it is measured in nanoseconds. Still a very fast process. Some factors can change how fast this goes. Something like temperature or pressure that's not mentioned in this particular diagram can change how fast the process goes. For this one, it just mentions the time, which is that T value up there, and then the capital T value over there is the excited state lifetime, also measured in nanoseconds. That's approximately how long those particular molecules will stay in that excited state, and it's going to vary just based on whatever molecule you're choosing. And then obviously we have intensity over here, which is measured in watts per meter squared. That's just the amount of energy you're going to have over a particular space. All right?